Welcome again. Why does sickle cell anemia happen more in some populations and less in others? Well, first let's find out what is the nature of sickle cell anemia. An individual who is affected by sickle cell has red blood cells or erythrocytes, which take on the shape of the crescent moon, very much like the sickle which is used to harvest crops, instead of the flattened disc shape. The reason for this is because of a gene mutation. A single base substitution mutation, which causes the alpha-globin polypeptide chain of hemoglobin to change its conformation. And when this happens, hemoglobin molecules stick together in low oxygen concentrations in tissues. Then there's a bundling or a distorting of the shape of the red blood cell as a result of this. This then leads to capillary damage, and the lifespan is much less than the typical erythrocyte. The generation of blood cells by the bone marrow does not respond to the shortened lifespan, so what it leads to is a permanent shortage of red blood cells. But when this sickle cell gene mutation arises, a single base in the sequence of DNA changes. Here on the sense strand of DNA, we can see how simple that change is, where a codon GAG changes to GTG. A single base, adenine, is replaced by another base, thymine. All of this, of course, on the sense strand. Messenger RNA then binds to this antisense strand, and it takes this GUG instead of GAG. Upon transcription, transfer RNA would bring the amino acid valine and not glutamic acid. And the result of this is an abnormal hemoglobin. Another way of looking at it is to say that this single base substitution mutation has led to the existence of a new allele in the population. And this allele is represented in this way: HB superscript S for sickle cell. And the allele that gives regular blood cells is represented as HB superscript A. With these two alleles possible in populations, then genotypes can look like this. The genotype that gives regular blood cells, like this. And then a very unique genotype, the heterozygote. Individuals that carry one allele for sickle cell and one allele for normal blood cells, they are described as having sickle cell trait, because in the case of this particular gene, the sickle cell allele is not recessive, but it exists as a codominant allele, meaning that it expresses itself in the phenotype or in the outward appearance of the organism, while not suppressing the other allele, which is the allele for normal blood cells. So the individual with sickle cell trait, the heterozygote, has some sickle cells and some normal blood cells. And finally, there's also the possibility of having individuals with 100% sickle cells, homozygous for the sickle cell allele. And it is important to note that this genotype provides an example of codominance, as distinct from incomplete dominance, which is a situation where one allele mixes its effect with another allele. A good example being red flowered plants being crossed with white flowered plants to give you pink flowers. But why is it that the process of evolution by natural selection has allowed such a seemingly negative trait to be propagated in the human population, to the extent that in some populations in East Africa, 35% of the population carry this genotype, and as many as 5% of newborn babies are homozygous for sickle cell. And there's a good reason for this. Individuals that have the sickle cell gene also have resistance to malaria. And if a population is affected by malaria, then this creates another factor in the struggle for existence. And individuals that are heterozygous will be best suited to win in this struggle for existence. Because owing to this codominance, they would have some element of normal blood cells and not suffer the debilitating effects of full-blown sickle cell, while at the same time having resistance to malaria.